Okay, so next one I'm going to follow with the last topic of this chapter, that is called genetic mapping. This is quite related with the linked gene cases. Okay, so first of all, you need to know what is the meaning of genetic mapping. Okay, so mapping that you are going to order the list of genetic loci along a particular chromosome. Okay, so the hypothesis here saying that the percentage of recombinant offspring, okay, reflected the distance between the genes on a chromosome. So it say that when the two genes are more far apart, the higher the probability that a crossover will occur between them. Therefore, higher the frequency of recombinant frequency. So for example here is a chromosome. So on this chromosome, there is several genes located at different loci. Okay, for example, we have gene A, we have gene B, gene C, gene D, gene E. Different gene determine different characteristics. So this gene mapping is going to locate where the gene is. Okay, where is the gene located at their respective loci. So the hypothesis here saying that when the gene is more far away, like for example A to E, then the higher the chance that crossing over will occur, compared to the gene which is very near to each other. For example, C and D. Okay, C and D is very close to each other, so most probably they are very hard to cross over when they form the homologous chromosome. Okay, so we can calculate the crossover value. Okay, it's called the COV, to measure the distance between the gene. Okay, you can measure the distance of A and B gene by using the COV value, crossover value. Okay, so it is also known as recombination frequency. So okay, this is the one I showed you just now. C and D appear very close together, therefore the chance of recombination is low. While E and F, they are very far apart, so there are higher chance in recombinant. Okay, so this one showing the gene mapping for different gene here and here. The value of COV, okay, very far apart. From here 0 to 48.5 to 57.5, 67 to 104.5, the distance. Alright, so there is a formula in calculating genetic mapping. So COV, crossover value, equal to the total number of recombinant divided by total number of offspring times 100. So when the value of COV is lower, means the gene is closer to each other. Okay, like the diagram just now. So when the value is higher, means the gene is more far. Okay, so therefore this COV can also used to locate positions of genes on a chromosome. So this is called chromosome mapping or genetic mapping. 1% of COV equivalent to 1 map unit. Okay, so sometimes we also use the unit of centimorgan. Okay, first example. Now you are given 4 genes, P, Q, R, S. So we know that P, Q, R, S are located on the same chromosome. So what you have to do is you need to map the chromosome. So we want to know the sequence of the gene. Is it PQRS or SRQP or QRSP or anything? So you are given the information as follows. The distance of P and Q is 35%. P and R 5%, R and Q 40%, Q and S 10%, R and S is 30%. So how to do? First we start with the furthest. Okay, furthest means they have highest COV percentage. So highest is 40% here. So R and Q is 40%. So first locate R and Q. They are 40 map unit away. So they are very far apart. Second one is P and Q, 35%. So P to Q is 35. So your Q is already located here, means your P will be here, which is 35 away. Okay, just now already 40. So 40 minus 35 means here is 5. R and S is 30. So just now we already located our R. So S will be somewhere here, 30. So means that 5 plus here, 25 equal to 30. Q and S is 10. So your Q is already here. So S just now we locate it here. So let's reconfirm. Is it 10%? Yes, 10% here. So now 5 plus 25 plus 10 equal to 40. So the location of the gene on this chromosome is RBSQ. Okay. So example number 2, you were given a test cross. Okay, a test cross. So now you find the number here, 300, 300, 100 and 100. So when you see two big numbers here, two small numbers here, so this is a case of linked gene test cross. Okay, remember you only can map the gene when they are linked gene. Okay, you only can map the gene when they are linked gene. Because if they located on the same chromosome, gene A and gene B, okay, then only you can map the gene. If gene A and gene B is on different chromosome, how do you map the gene? You do not know the distance with different chromosome. So now, since it is a test cross, okay, so we know that we must have two parental genotype and two recombinant genotype. So from these four numbers here, the recombinant genotype usually have smaller number. Okay, so we use the formula COV equal to total recombinant divided by total offspring. Okay, offspring times 100. So now total progeny is 800. 300 plus 300 plus 100 plus 100. So total offspring is 800 here. Total recombinant 100 plus 100 times 100. Okay, the formula. So now 25%. 25% means what? Means the two genes S and M is 25 map unit away, because 1% equal to 1 map unit. So you map the gene S and M on the chromosome. Okay, on the chromosome S and M is 25 map unit away. Alright. So example 3, you are given a test cross. The results from the test cross, again two big numbers, two small numbers, means this one is parental genotype. This is called recombinant. So map the gene of C and S. So use the formula COV equal to our parent recombinant. Total is 100. So COV equal to total recombinant divided by total offspring equal to 4%. So 1% equal 1 map unit. So it's 4 map unit away. Okay, means that your C and S gene, the C gene and the S gene is 4%, 4 map unit away. Okay. Okay, so the last example here, another sets of the linked gene test cross, 2 big numbers, 2 small numbers. So the first question, what can you conclude about the gene? So you say that the 2 genes are linked, located on the same chromosome. Reason, because the ratio from the test cross is not 1, 1, 1, 1. So calculate the map distance between the gene. So only linked gene can calculate the distance. So we use the formula here, distance equal to total recombinant over total of offspring. So how do we know which one is recombinant? The smaller number here, 403,397. Divide by total offspring times 100, so it's equal to 9.09%. 1% equal 1 map unit, so 9.09 map unit. Means that the gene for flower color and the gene for pollen grain shape is 9.09 map unit away. Okay.